Hi everyone, Brenda Gustin here, and we're on section 1.4, naming, classifying, and identifying microorganisms. Those three things together, naming, classifying, and identifying, are what we call called taxonomy. And so down here it says the primary concerns of modern taxonomy are naming, classifying, and identifying. When we have living organisms, scientists always want to put organization to them, and it helps us understand them. It helps us study them more effectively. And so the study of taxonomy is giving things names, putting them in some kind of orderly um, organization. And then sometimes we need to identify them. We need to, find something out in a natural environment and say, what is it? And so we should have rules for how to do that, both given for all of these. And so I just wanted to touch on these, and they're important in microbiology. One of the things, let's see, one of our learning outcomes is correctly write the binomial name for microorganism. I have a bird feeder outside, and I can look out there and I can recognize a rose-breasted grosbeak and a grackle and a dove and who knows what else is out there. And I have those common names. But when we look at bacteria, they all, a lot of them look a lot alike, and they don't, nice, cute little friendly names don't pop into your mind. And so we typically use the scientific names. Now, that doesn't mean that a rose-breasted grosbeak doesn't have a scientific name, because it does. But we tend to use the scientific names for bacteria much more frequently than we do for a lot of the things that we see day to day. And so the method of assigning the name, or the way that we give living organisms name, is called the binomial system of nomenclature. Binomial two name system of nomenclature. And what you see is in the binomial system of nomenclature, we have a two words to give us a name, and probably everybody's heard of E. coli, but its real name is Escherichia coli. So we have the two names there. Escherichia is the genus. And so the genus is going to be the first name in the binomial name, or in the scientific name. Coli is going to be the second part, and that's the species. Now, these names don't just pop up randomly. They don't just pull one out of the hat. They come up with things. And it turned out a guy named Eschrich, he discovered Escherichia coli. So it was named after him. I think he named it after him. And he found it in the colon of people. So they kind of Latinized colon and put coli. Here's a little story about a tree hopper, an insect that was named after a little girl. So her name was Sylvie, and they kind of Latinized her name a little bit, and they ended up naming it after her. I forget what they named it. Where'd it go? Oh, anyway, it's in there somewhere. And so here you can come up with these binomial names for living organisms. And so every living organism has that. So when you're writing your scientific names, notice up here, here's a lot of scientific names. The genus, the first word, is written with a capital letter at the beginning. And so Neisseria gonorrhea causes gonorrhea. Saccharomyces cerevisiae is brewer's yeast. Saccharomyces is a genus, it's capitalized. Here we have Neisseria gonorrhea. Neisseria is the genus, it's capitalized. Gonorrhea is a species, and it starts with a small letter. So you always write it like that. And then notice also that these are italicized. Now, if you're going to put it in, use a typewriter, use a computer to write it. If you're going to type it, usually we italicize it. Whereas if you're going to handwrite it, we usually underline it. And then once you've said the name once and people know what you're talking about, you can abbreviate that. And the way you do that is you take the genus, you take the first letter of the genus, and you put a period. So you can turn that into E. coli. And you typically would do that after you've heard of it, after you've mentioned it one. N. gonorrhea, A. Baumannii, I'm sorry, A. Baumannii. Bowman is the person that discovered that. Let's see if there's here's some examples. Oh, wait. Lactobacillus like San Francisco, and they come up, their names come from all different kinds of things. Here, this one says it's a bacterial species used to make sourdough bread from San Francisco. Staphylococcus aureus. Staphyol is Greek for a bunch of grapes, and coccus is berry. Aureus means golden, and if you grow Staphylococcus aureus on certain kinds of media, it has kind of yellowish colonies. So that's nomenclature. So you should be able to write those names. In fact, I have a practice question that asks you to write the word Escherichia coli 4 different ways with correct scientific, using correct scientific writing. Let's say classification. So taxonomy is nomenclature. How do you name it? Classification. And probably most of you, again, are familiar with how living organisms are classified. And we have the kingdoms. Oh, remember, we actually started with domains. We had 3 domains that we talked about in section 1.1. See if you remember what they are. Bacteria, archaea, and eukarya. And so, and then within, here's some organisms within the domain eukarya. We have kingdoms, phylums, class, order, family, and then down here's where you get the scientific name, the genus and the species. And so what you're going to notice is that these organisms are grouped based on similarities. So let's say here we'll start. Well, let's do a microorganism. It's Paramecium caudatum, and so it tells you some things about this certain kind of protozoan, and it's in the family that has cells that rotate while swimming. So cells that do that are all in the same family. And then that can get into a broader group called an order. And the order, these are elongated cells with cilia. And then we can put it into a larger group called a class. So all the organisms that have these characteristics, the characteristics of single cells with regular rows of cilia are in the same class. And we can keep broadening that out. And here we get into the kingdom Protista. And get all the way down into the genus and species. Now, when you look at these, we say, how did they group these organisms together? Well, we know that, the protists and the algae, they're the single-celled plants and animals. And so those single-celled plants and animals that have cilia are in the Coeliophora. Excuse me. And we keep narrowing that down. And this is typically how we've done it. Sometimes they don't just look at visual characteristics to put them into these groups, but they might look at metabolic characteristics or different kinds of way they feed and things like that. So historically, we've been able to classify organisms into these different groups using those kind of characteristics. The last learning outcome says explain the difference between traditional and molecular approaches to taxonomy. So what happened is, I'm going to go down here, is it says in 1975 there was this guy named Carl Woese, and he started studying certain kinds of RNA and that all cells had. And he found that you could kind of analyze the RNA, the sequence of the RNA, and determine how closely an organism was related to others. And you can use that RNA to identify organisms. So we call this a molecular approach to identifying organisms or finding their relationships. And so it goes into a lot of detail here, more detail than what you really need to know. But the bottom line is, in about the 1980s, instead of just classifying things based on what they looked like or based on their metabolism, we actually can analyze their RNA and look at them molecularly, look at their molecules, and much more accurately say, how are these things related? Which ones are actually more closely related? hippopotamuses and pigs look a whole lot, they kind of look alike. You might think they're related. But when you do molecular analysis, so you look at their RNA and their DNA, it turns out they're not very closely related. And so when we started doing these molecular analyses in from 1975 on, it completely changed how we look at taxonomy and how things are related. In fact, this is what led to the 3 domains that we talked about in section 1.1. Remember, we said we've always classified organisms into 5 kingdoms, but once we were able to analyze things at the molecular level, we realized, gosh, we really had some things wrong. And a big one was bacteria and archaea kind of look alike. So we put them all together in one group, but now we know they're really genetically distinct and they're in 2 totally separate domains. So, little note here, it says, please note that viruses and prions are not included in any of the classification systems because they're not living organisms. They're not cells, they're not living organisms. And that's all I'm going to talk about on section 1.4.